Welcome to the August 2025 edition of DataBytes. Today we're introducing the NCATS Biomedical Data Translator, a new tool for biomedical data integration and inference. I'm Amanda Miller, and I'm the Associate Director of the National Consortium for Data Science, or NCDS, a project of the Renaissance Computing Institute, also known as RENCI, a research institute at UNC Chapel Hill. Before our host and speaker begin, I just want to share a couple of housekeeping notes. Today, the chat feature will be open for attendees, and we will be using the Zoom Q&A feature to manage our questions after the presentation. Immediately following our Q&A, I'm going to take a moment to tell you about a few upcoming NCDS events. So I did mention earlier that NCDS is a project of RENCI at UNC Chapel Hill. And now I'm going to pass the mic to RENCI Deputy Director Becky Boyles, who is going to be introducing our speaker and topic for today's DataBytes. Becky?
Thanks so much, Amanda, and good afternoon, everyone. Welcome to another edition of NCDS DataBytes. Today, we're exploring cutting-edge developments in computational data science that are transforming research across disciplines. As Amanda said, I'm Becky Boyles, Deputy Director at RENCI, and I'm thrilled to host today's session. At RENCI, we're deeply committed to advancing data-driven discovery through innovative computational approaches, collaborative research, and the development of infrastructure that enables scientists to tackle complex challenges. Our work spans multiple domains, but today we're going to focus on the most promising frontiers in computational biology and translational medicine. I'm excited to introduce Chris Bison. He's RENCI's Director of Analytics and Data Science, and he'll be presenting the Biomedical Data Translator. This is a groundbreaking knowledge graph system that has some exciting capabilities that will change how users integrate and make sense of biomedical data. Chris will walk us through how this open source platform is breaking down data silos by harmonizing diverse biomedical data sets and enabling powerful inferences that can accelerate discovery. Through a few compelling real-world use cases, Chris will demonstrate how Translator is moving us closer to a future where computational approaches can directly impact human health outcomes. This work exemplifies RENCI's mission to bridge the gap between computational innovation and real-world applications in order to benefit society. We hope this presentation will inspire questions and comments, but we ask that you hold your items till the end and utilize the chat in the Q&A function when we open that at about 40 minutes. I'm looking forward to moderating a lively Q&A. And without further delay, I want to hand it to Chris to talk about the Biomedical Data Translator Program.
Thanks, Becky. Just going to share my screen here. Hopefully I've shared the right thing. Are people seeing the slides?
Looks great, Chris.
Great. Thank you, Amanda. So as Becky said, my name is Chris Bison. I am the Director of Analytics and Data Science here at RENCI. And I'm going to tell you about a project that we've been working on for a number of years now, the NCATS Biomedical Data Translator. This is a large consortium spanning researchers from somewhere between 15 and 18 different institutions. Over the years, there's probably been, up to 100 participants. The particular slides that I'm going to show you are slides that are also contributed to by a couple of people, Andy Kraus and Kara Fecco. We have recently released version 1 of the translator, and I know at this point you're going, I don't know what you're talking about. What is this translator thing? And I'm going to show you in a second. But I will just point at this paper that has just come out in Clinical and Translational Science for a couple of things. One, because it is the record of the release. It explains all about Translator, what it is, how it works, and so on. And it also contains the full list of collaborators on the project, including a very healthy list of consortial authors. So as part of the publication process for this release, we have created a little video about what Translator is and how it works. I'm not going to play you the whole video, but there's a couple of minutes that I especially like. So I'm just going to pop out of the slides here for a second and move over to the video. And I'm just going to play you this little story about how Translator can be used that hopefully sets the scene for the rest of the talk here.
A 10-year-old girl began inexplicably vomiting every day after returning from a family vacation to Europe. For weeks, doctors assumed her symptoms were stress-related until she was suddenly hospitalized with copper poisoning and liver scarring. After extensive testing, her clinical team discovered that a high copper diet of seafood and nuts during the trip had triggered the progression of an undiagnosed condition, Wilson's disease, a rare genetic disorder caused by mutations in the ATP7B gene. ATP7B helps to eliminate excess copper from the body, but mutations in this gene disrupt that process and cause copper to accumulate in the liver and brain, leading to symptoms that can range from neurological issues to liver failure if copper levels are not managed. After her Wilson's diagnosis, the patient's siblings were tested, and her 2-year-old brother was found to have the same gene mutations, despite being pre-symptomatic. These findings raised a series of questions for the clinical team. What drugs can manage copper levels and prevent the progression of Wilson's disease? What drugs might increase the activity of ATP7B to improve copper elimination in the body? Are there any clinical trials that support the use of these proposed drugs in managing Wilson's disease? Biomedical researchers can use Translator to approach these questions by asking, for instance, what drugs may treat conditions related to Wilson's disease? Translator returns a list of potential treatments, with trientine hydrochloride appearing as the top result. This drug is already used in the treatment of Wilson's, so Translator's reasoners correctly ranked it high in the results. Expanding the result shows a series of paths formed by the hundreds of data sources Translator uses, demonstrating the relationships between the drug and disease. Some of these paths show a direct connection between them, while others use multi-step logic involving intermediary genes, phenotypes, drugs, and other biomedical entities to form a connection. Clicking on a relationship within a path reveals supporting evidence, including publications and clinical trials. For further relevance, researchers can filter for trials with pediatric indications or that are actively recruiting. If trientine hydrochloride isn't a good option for the patient due to her age, drug intolerance, or other concerns, Translator's reasoners suggest over 1,000 other drugs that could be used as possible treatments.
Right. So I'm going to pop back over to the slides now. So hopefully that gives a rough idea of the sorts of problems that Translator is meant to address. And a very, very brief introduction to the tool itself and the UI there. So having given you some idea of what Translator is, that is, it is a tool that integrates a bunch of biomedical knowledge and exposes that knowledge to translational researchers in an effort to help them answer questions. I'm going to dig now into the weeds and start talking a little bit more about how it works kind of from the ground up. So we'll go all the way back to the beginning and sort of build Translator up from scratch, and then end up back sort of where we're starting now, and talk a little bit about, again, sort of what it can do, what we want it to do in the future, and how you can get involved. Okay. So I'm going to go way back. I'm going to go all the way back to this guy. If you don't happen to recognize him, this is Carl Linnaeus. And this is essentially the founder of modern taxonomy, right? I think that biologists, life scientists in general, tend to be very good at systematizing things. And this person is an excellent example. He developed the system that we all learned in, I don't know, middle school biology or something. You learned kingdom, phylum, class, order, family, genus, species. You learned about binomial naming of organisms, right? So genus, species is how we often refer to things. And you know, in some sense, it feels like, well, okay, he made a tree and he put some stuff on a tree, but it's actually a huge achievement, and it led to him being really a scientific rock star in the 1700s, because you know that system, that structuring the data in that way allowed people to take this huge diversity of organisms, plants and everything, you know, animals and fungus, and figure out a way to build a system that you could really understand. This kind of duck is the same as that kind of duck, but it's different from this other kind of duck. And this is how these things are related. And these are the things that are more closely related, and these are the things that are less closely related. And these are how they differ. And biologists have continued along this systematizing effort all the way into the modern times. And now when we talk about how do modern life scientists organize knowledge, one tool that is very frequently used is this thing called ontologies. And ontologies are, again, a system of organizing facts or information or entities. But these schemes are ones that are very amenable to being used computationally. So one of the most famous of these is the gene ontology, or GO, which has been around now for over a couple of decades. And essentially, it is a way of systematizing information about all the things that genes can do, right? So genes do an awful lot of things, proteins. And for instance, a protein may be involved in a process like ATP binding. Well, ATP binding, that's a very specific thing. You know, you're binding to this very particular molecule. That's actually a subset of a more general function, which I am not sure I can read, adenylribonucleic binding, which is itself a subset of a more general function, purine ribonucleic binding, which is a more general, a more specific form of a more general, all the way up to, you know, a very general term like binding. And so that's for gene functions. You can have similar kind of hierarchies of knowledge around things like diseases. So here's a little section from a disease ontology called the monarch disease ontology. But these ontologies can span a range of different types. So you can have what genes do, you can have diseases, you can have disease symptoms, you can have cell types, anatomical features, all of the components of biomedical knowledge can be systematized in these ways. Okay, so what? This enables you to now start to say very precise things about these entities. So we've got maybe one classification scheme talking about chemicals, and we might have a chemical like ozone, and that ozone has an identifier in that vocabulary. And maybe in a disease ontology like Mondo, we have another term for asthma. And we have the ability now to make these pretty precise statements that are sort of subject, action, object, like ozone exacerbates asthma. And we call those triples. And those are really, in some sense, the sort of little Legos of knowledge that we're going to try to click together in order to form explanations or stories or reasoning. When we take a whole bunch of triples, or a whole bunch meaning a few, but up to, say, 100 million, or somewhere in that range, and we put those triples together, we form something called a knowledge graph. So this is a picture of a small knowledge graph. Here, graph should be understood in the sort of computer science meaning of the word, where it's a structure where you have nodes, and here those nodes represent biomedical entities, and edges representing the relationships between those entities. So here in this particular graph, what we're looking at are our nodes might represent a gene like this DDHD1, or they might represent a process like lipid metabolic process or a chemical like pyruvate. And then the edges represent the relationships between those things. So RS1954322 is a variant nearby the DDHD1 gene. And by looking at the right section of graph, you can try to understand, what's happening biologically in a little bit of the world. Okay, so we have biologists out there creating all kinds of biological knowledge and systematizing it into these machine-readable, understandable little chunks of knowledge. Great, awesome. Now, all of that knowledge, where is it? And how do we get at it and do stuff with it? Well, the other thing that's happened a lot, right, is different people take these pieces of information and they put them in databases. And there's no plan, right? There's no master plan for who puts what where. It's just, person A figures out some stuff or collects some information and they make a database and they put it somewhere and they say, here's my database. And person B gets another, little bit of data and they take it and put it together and systematize it and make it nice. And they put it out there and they're like, here's my database. So we end up with all of these little databases all over the world, right? And they all contain little keyholes that you can look through to see little bits of information. So we may have over here a database that contains all kinds of stuff about drugs that bind to genes. And maybe somewhere else, we've got another database that's got all kinds of information about how genes are involved in diseases or which diseases involve which genes. We have other databases, these are where genes are expressed, which tissues they're expressed in. How can we use all this? Okay, so now we've got all this systematized knowledge. People have made it available, but it's kind of scattered. And it's not really easy to take advantage of all of this work, even though people have made it available and they've made it at least structured. The real issue, there are a couple of issues. One is that because, again, people have done this just individually, there's a lot of variation in how it's done. So I said that biologists love to systematize things. Part of the issue is they love it so much that they've done it over and over and over again, even for the same concepts. So if you think about something like genes, there are not just one way to describe genes or give genes names. There's actually a bunch of them. So there's the NCBI has a list of genes and they've got, here's all the genes and they've got NCBI identifiers. There's another group, Ensemble. They made another list of genes with Ensemble identifiers. They're the same genes, right? Because there's only so many genes, but they're now can be described with two different sets of identifiers. And if one of my databases used one of those sets and another database used a different set, I now have work to do if I want to go try to use those together. And I do want to try to use those together, right? So if I am a biomedical researcher, maybe I'm a bioinformatician or translational researcher, I think there's a lot of value in this data. So for instance, I might be interested in doing something like drug repurposing. In drug repurposing, you may have a disease that you're interested in. It's very hard, obviously, to come up with a new chemical to treat that disease. The idea of drug repurposing is, well, maybe I can find a chemical that's already used, it's already approved to treat something else, but maybe it will have salutary effects for this disease that I'm interested in. And I may have an idea for how to find such a thing. If I said, well, if I took my disease, let's say I'm interested in Castleman syndrome, and I said, what genes are involved in Castleman's, and then what drugs do I know that interact with those genes, right? That's a fairly straightforward query that you could imagine wanting to run. It won't answer your question completely, right? But it will give you a set of hypotheses from which to work. But assembling even that sort of two-hop set of data involves at least two different data sets, usually multiple different identifiers, and an awful lot of work. And so people kind of either push through and spend a lot of time doing data cleaning and data integration, or they give up. And this is one thing that the data translator is there to fix. So we're taking all of these different data sets that are out there and making them conform to a common model, use a common set of identifiers, and make all of that available to answer questions for people. I'm going to keep moving. So having kind of now built up from data principles up to the idea here, I just want to talk a little bit about the goals of the project. As I said, one of the goals is just to unify access to this big range of biomedical knowledge sources that's out there. And not just unify access, but really be able to integrate the data together. Because until the data really is put together, you may almost as well not even have it. That's all great, but there's more to what Translator wants to do. Part of what we want to do with Translator is not just provide a query engine or a Google for this kind of data, but actually use it to perform reasoning and provide hypotheses to the user that go beyond just reporting exactly the facts. And finally, it's extremely important for Translator to be able to show its work, especially if we're making an inference. The idea is we're not just going to say, well, our computer predicted this, so here you go. Instead, what we want to be able to do is to say, okay, we're suggesting that X drug might treat Wilson's disease. Here's why we think it, right? Here are the pieces of evidence that lead us to that belief. And here are the sources of those pieces of evidence, all the way back to papers or whatever the original source of data is. So here's the architecture of the tool that we've built, and I'll walk through it kind of forwards and backwards, I think. At the top is the UI, and we'll take a look at the UI. When you choose a question, that question goes through a little bit of routing, and it ends up at these things called autonomous relay agents. Their job is really to answer the question. They can go out and talk to translators' knowledge providers. The knowledge providers are the tools that have ingested all of these knowledge sources and made them really interoperate well together. So the UI asks the question, it gets to the ARAs, they go out, they get data from the KPs that have previously ingested the knowledge sources, they provide the results back to the ARAs, which synthesize things up, until it finally gets back to the user. What makes all of this work is internally a set of data standards that allow the same question to be posted to different tools that may have different data types in them or different mechanisms for how they're going to try to answer the question, but they all speak through the same APIs and are able to pass data to one another without worrying about the internal details of the tools. We do have some fairly fancy tools for handling this identifier problem, which, depending on who's on the call, is the kind of thing that maybe about, 95% of you are going, who cares? And 5% of you are like, yes, 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 finally. But we have a tool that has ingested, all of these different vocabularies and done a lot of work to try to figure out, oh, these are actually the 15 different ways that people refer to water. So whichever one you find, we know we're talking about the same molecule. I want to talk briefly about how these questions are answered. So I said that there's these tools here in the middle, the autonomous relay agents. These are the individual components in Translator that get a question like, what might treat Wilson's disease, or what might upregulate MEC or something. And they figure out how to go get the data from the knowledge providers and answer the question. Part of the goal of Translator is to provide an array of different methods at this layer. So the idea is not, oh, Translator always does it this way, or this is the Translator method. Instead, there are multiple components in there, each employing different methods, a sort of a diversity of methods, that then those answers are all integrated together, and we can say, oh, you know what? I got this answer from both method A and method B, so that gives it a boost, and we can say we arrived at this answer through multiple approaches. So a couple of those approaches, for instance, might be rule mining. So the idea of rule mining is I've got a big knowledge graph, and I'm going to learn statistical patterns in that graph. I maybe learn that if I have a drug to gene to disease path, that tends to be correlated with a drug interaction with that disease, right? That would be a rule that I could then go apply if someone asks me to find a certain path. I can say, oh, these are the other patterns I can go query for to see, to make a suggestion of an answer. Or there are other tools that are taking more of a machine learning approach, where you take your knowledge graph and you convert it all into a series of vectors. Once you've converted things into vectors, then you can run off and do all your great, neural networks and, random forests or whatever kind of machine learning approach you want to take in order to make predictions. So these are not incompatible strategies. They are, other attempts to answer the same question that we can fit into slots in Translator and benefit from. Okay, so that is a bit about how Translator works. I'll go through some examples of, if you go to the UI, which is there, ui.translator.io. Translator is spelled a little funny, so you got to be careful if you're going to type it in. And show you a couple of, what you would see here. So the back end, as I've told you, is quite general, right? It has this very broad way to query a wide array of data sets. And we have tools for reasoning over that data. What is exposed in the UI is sort of a subset of those things, which is a specific set of 5 queries that we know that the reasoners are capable of answering well right now. And those are what drugs might impact a condition, and then a number of questions related to the interactions of chemicals and genes. So what chemical might upregulate a gene or downregulate it, and conversely, what genes might be up or downregulated by a chemical. And again, the goal here is not merely to provide an answer that is known, but also step outside the box, use that information that's in Translator to try to make further hypotheses for the user. So for instance, I might have a question like, I have a drug, celiprolol. I want to know what genes it decreases the activity or the abundance of. I might find some direct answers. Maybe there is a binding assay, or maybe there's some RNA-seq data somewhere or something like that. And I get a direct answer to this question. And it's based on, an experiment or a paper or a fact of some type. But I also might have pathways like this, where I have a fact, or at least a statement in a paper, that celiprolol increases the activity of this gene, gene A here, and that I know from another source that gene A decreases the activity of some other gene, gene B. Well, I can then infer that celiprolol will indirectly cause a decrease in the activity of this downstream gene, even if it's never been measured, right? Now, I might be wrong too. So it's a prediction, it's a hypothesis, but it is potentially helpful to a user to be able to see this, especially if we can provide them the detailed information about these known edges and they can decide, is this a valuable thing to follow up on. So here, if you put in a query somewhat like that, are the kinds of things you would see. Here we have a couple of direct edges. So here's celiprolol. Here is a gene that is having its activity decreased by celiprolol. And what we're seeing here are direct relationships between the two. And there are two of them, actually. So it kind of keeps separate individual statements so that you can look at each one. And if you click on one of those, it'll bring up what the supporting evidence, like why do we think that edge is there? And that has a couple of pieces of information. One is, where did we get it? What was the primary source? What was the first place that this statement appeared in a database somewhere? And in this case, that is DrugBank, which is a well-known resource. And then DrugBank itself may provide also their evidence, usually in the form of a publication. So a lot of the databases will do this, especially if it's a curated database. It may have a statement like, celiprolol down regulates ADRB1, and they may have in their records that there's a particular paper. And so we carry all that along, and we can say, okay, here's the publication. You can go click on this. It'll take you to PubMed. You can read it and decide, is this right? And then over here on the right, we see an example of one of the more indirect cases where we're saying celiprolol will decrease the activity or abundance of COL1A1, but you can see it's got this like dotted line here. And what that is indicating is there's not a direct evidence. There's no paper that shows this exactly. Instead, we are finding paths that are somewhat longer that are, oops, sorry, my computer just did something odd. Nope. That are supporting that. And we can, again, dig into the individual pieces. So here we're saying, oh, celiprolol is increasing the activity of this gene, which is decreasing the activity of that gene. And I can go into each of those things which are direct edges and find out about their provenance and follow up on them. There are other things built into the UI to help you dig through answers. And if we have time, I'll look at a live version of this. But for instance, here's a different question where we've asked what might increase the activity of MET. And there's a drug that's come out. We have here a bunch of all of the supporting paths, which again, these are like these two-hop sort of statements that are leading us to believe that there's an indirect relationship here. But the UI also gives you some filters. So you can see, for instance, some information about this drug up here. So you get a description of what the drug is. And then you get some roles, classes that this drug is in. So this is an anti-neoplastic agent. And you can see how many other anti-neoplastic agents are found by translator. So you can use these to like filter and say, well, I want to see more of these. I want to see fewer of those, and dig into the results that way. So those are the things that are currently in the translator interface. They are being worked on still and improved. But we're also, over the next funding cycle, going to be broadly expanding the number of things that translator can do, right? So in addition to these sort of canned queries that we're going to continue to develop, we have two more general queries that are currently in development. One is called Pathfinder. And the idea of Pathfinder is, well, let's say I have an observation. I observe that two genes are co-regulated in some system. I would like to just understand, what is the relationship between these two genes? Maybe they have some upstream chemical regulator, or maybe they're part of the same pathway, or maybe they're part of the same disease or something else, right? And so the idea of Pathfinder is to give you two ends, right? I'm at home and I want to go to work. How do I get there? Google Maps, right? And you'll find a path through the graph that hopefully is explanatory of whatever observed connection you have between those two entities. So that's Pathfinder. That's an interesting project. Really, the tricky part there is not finding paths. When you take this much data and you put it together, essentially everything's connected to everything. The really interesting and difficult part is, well, how do you find paths that are meaningful, right? Another capability that we're working on quite strongly now is using enrichment within a graph. So the idea here is somewhat similar to Pathfinder, but instead of saying, I have two things, find me kind of a long, snaky path through the graph that tells me a story, instead, the question is, I've got a whole bunch of things. What do they have in common, right? So for instance, I might have a set of phenotypes. So I've got a person. They have a series of symptoms, and I don't know what disease they're suffering from. Maybe this is a rare disease, and we're kind of in the midst of a diagnostic odyssey, trying to figure out, what is this person's diagnosis? In our knowledge graph in Translator, we have a bunch of symptom, or here called phenotype, to disease relationships. So if I have a bunch of phenotypes that have been measured in this person, I can then ask, well, what diseases are preferentially connected to that set of phenotypes within my graph? And then that provides hypotheses about what the potential diagnoses of this person could be. But the nice thing about this approach is that it's completely agnostic to the types, right? So you could go from symptoms to diseases, but you could also, using exactly the same kind of formalism, say something like, I have a bunch of blood metabolites that are differentially measured in this person. What genes seem to be the kind of controllers of this set of metabolites? Or, I've got a set of genes from, I don't know, an RNA-seq or something. What pathways are they mostly in? So kind of any sort of A to B kind of combination that you like can be queried in Translator to give you these sorts of answers. One other kind of project that's in dev is sort of integrating the kind of knowledge graph reasoning with LLMs, which of course is not a surprise. The way we're approaching it now, right, is we have the data, right? We're comfortable with our ability to ingest and structure all of the information in a way that is relatively clean. When we show the UI to people, right, I would say there's a lot of people that are like, great, I get this, I see what's going on. And then there's a fair number of people that are like, oh, what are all these like boxes and lines and wiggles and stuff? Just tell me what this means, right? And so what we're working on is the ability for a large language model to be able to consume the structured output of Translator and summarize it in text for somebody. So here's an example where I've asked Translator for drugs that might impact PTSD, and I've asked it to summarize the result for fluoxetine. And if you look through this, it basically does a decent job of saying, well, there's this paper, there's not a lot of evidence for this. There is a paper, there's also a clinical trial, and then there's some subgraphs that are going through genes. Those genes are definitely related to other psychiatric disorders, so this kind of makes sense, but it's hardly a smoking gun. And it provides, I think, a fairly nice summary. So these three tools are things that we'll be rolling out hopefully over the next 6 months to a year. I am approaching the end of time, but I want to go through one more thing that hopefully will be interesting to people. And that is, as we're going through our release here, we're definitely doing some community engagement. And we have a program that we're launching now for translator ambassadors and champions. And what are those? Well, the idea of an ambassador is a clinical or translational scientist who wants to engage with the translator system and use it to advance their individual research. And these are kind of the main users of the tool. So people that would be using the user interface to try to answer questions that are useful in their research. The champions are a little more the software developers or the semantic engineers or the bioinformaticians who would be interested in either developing code or using kind of the back-end systems of translator and writing, more code to query those directly and do more complicated analyses that maybe are not available through the UI. Maybe taking some of those analyses and developing them into things that would be exposed through the UI. So there's a couple of different roles and ways to engage there with that program. So there are, the benefits, like what would people get out of joining a program like this? There certainly is a training aspect. So we would help people, our translators and, our ambassadors and our champions. We get training on how to use the tools, how to query the back end, a lot of networking with the consortium and with the NCATS program leadership, which is heavily involved in the program. Scientific authorship and paid travel to a local or national conference. Our hope is that people join the program, they use the tool, they make discoveries, and then they use those discoveries to both promote their own research and the translator tool. If this is something that sounds interesting to you, there are a couple of ways to get in touch. There is an email here, roverrelations@lists.renci.org. You can write to them and say, hey, I saw this talk. I didn't totally get it, but it sounded interesting. Tell me more, right? Or if you're like, yep, I got it, I'm on board, I want to do it, there's a link here you can click to go to this Google form where you can apply to join the program. It's kind of a rolling thing. We're going right now. We have our first tranche of ambassadors and champions that are being onboarded, and we're learning from that group how this will proceed. So if you joined up, you wouldn't be the guinea pig. You'd be in the second tranche, which will start, or the enrollment period will be December and January. But I think if you were really excited about it, you could go ahead and fill out the form, and it would be waiting for the people to review in December. I believe that is the end of my slides. I thank you for your attention, and I'm happy to take any questions that people may have.
Thanks for that, Chris. We have a couple questions in the Q&A, so I'm going to try and read them out for the sake of the recording. So the first question is amazing work, and thank you for hosting the webinar. First question is, why does the system design benefit from multiple KPs and ARAs? Why not convert requests to queries on the graph directly? And then second follow-up question, what graph query language is the system using in the back end? Is it a Cypher-like or something else?
Mm-hmm. Yeah, great questions. And I would say great questions with somewhat complicated answers. The part of the idea of the initial setup was around each of these tools was really being funded by a separate grant. And the idea for the KPs was really, well, what we'll do is we'll fund experts in this kind of data. So there would be one grant that was working all about, genetics data. And they are the world's experts in genetics data, and they're going to get the ingest of that all exactly, beautiful. And then we'll have a team working on chemical data, and they're going to get the chemical data just right. And so we would have these, kind of little islands of knowledge that we could then integrate together at runtime. On the ARA side, which is the reasoners, the methods for answering the questions, it's a little bit slightly different answer. The original idea there was to provide a diversity of methods. So if, as I talked earlier, there are teams that took a kind of a rule mining approach versus more of a machine learning approach. There are a lot of different ways you could try to answer these questions, emphasizing maybe different data sets or emphasizing different approaches for different types of questions. I would say over time, we have decided we like the different sets of ARAs. That is we like the diversity of ways to answer the questions. The diversity of knowledge providers, as opposed to putting things into one big platform, is something that we're kind of rethinking a little bit right now. There is actually a lot of value in a modularity there. For one thing, it makes it much easier to maintain. So if you have, let's say you're interested in building a big knowledge graph, right? Everybody's first attempt at building a big knowledge graph is to take all these sources and just dump them in as you read them, dump them in. Now you want to go change one source. Oh, I found a bug in this source. You don't have a modular ingest. You've got now one big thing that's made from a whole bunch of stuff, and you want to change one little bit of it. And it's fairly annoying to update that. So what's much more useful is to individually parse individual sources, into little chunks that are now very nicely interoperable with all of your other little chunks. And at that point, you have a choice, right? You could take each little chunk and stand it up on its own, and at query time, know exactly where to go. Or you could take all of them, smush them into one big thing, put it somewhere, and just pound on that. In the current phase of Translator, which we have just started, we're moving into a system that kind of allows for both. So we're going to have this series of individual sources, and at the same time, one big graph made from the different sources all smushed together. The reason there for maintaining both is that they really serve different needs. The individual sources can be more comprehensive, more frequently updated. The big knowledge source, it may be hard to take all of those individual sources and get them into one graph. The other thing is, a lot of times when you build one big graph, it's good for some things, but not other things. So it's really nice to be able to have the flexibility at various times to take your set of individual pieces and combine them in different ways, right? To provide different views that have different features. So for instance, just a very goofy thing, if you've got, say, a bunch of diseases in there, do you want all of the subclass edges between those diseases? Is that useful for your purpose? Sometimes yes, sometimes no. Okay, let's say you do want those. Do you also want all of the inferred subclass of edges? So if you have A as a subclass of B as a subclass of C, you can infer A as a subclass of C. Do you want that in your graph? Well, sometimes you do and sometimes you don't. So in making a big graph, there's always a set of choices about how to do it. By maintaining all the little graphs, you can build whatever big graph you want when you want it for what you want it for. So that was a somewhat long-winded answer, but I think hopefully modularity, right, is always a good thing in software design. Mostly always. Now, the other question about what is the back end, again, because the initial idea of translator was really to have all of these different components, and as long as they all spoke the same language, we didn't care how they were implemented. So the current knowledge providers kind of are a mix of things. There are some that are sort of a Neo4j kind of cipher thing. There are other ones that are really SQL backends that know how to kind of transform the graph query they get into their SQL queries. The communication layer between the different tools is a JSON-based graph query language called Trappy that we developed for the program. As we move towards unifying the backends now, our big graph will probably be cipher based, but it's unlikely to be a Neo4j container for reasons that I can go into if you're curious.
No, that's great, Chris. Thanks for adding that. Another question somewhat related. So two parts. Does Translator have an API to access? And then how flexible is Translator to extract a sub-knowledge graph of interest and transform using your own graph algorithms for downstream analysis or prediction?
Yep, those are great questions. The first answer is yes. Translator has APIs basically at every one of those components that I talked about. So ARAs have APIs, the ARS has an API, the node normalizer has APIs, the individual KPs have APIs. So it's very flexible in how you query it and what you do with it. Honestly, if you're really excited about using those APIs, the best way to learn about them is to become a champion, and we'll train you up on all of that. There is documentation as well, but it is moderately complicated. So absolutely. Now, in terms of pulling the graphs and taking them downstream, I would say the current ability to do that is somewhat limited, but in about 6 months, it's going to be great. That is, in the current, I talked a little bit about this sort of re-implementation of some of the back-end pieces to create some larger graphs and unify some of the back-end components. Part of that is going to be creating some, cloud buckets for storing graphs in a way that we don't currently have. Those are all going to be available for access for people to pull. The queries right now, it would be harder to, it depends how big of a query you want to pull, how big of a graph you want to pull out. If you're talking about bite-sized things, the current query mechanisms will do that well, and you could take them for downstream. If you're talking about, I want to get, a very sizable chunk of the translator graph, that's a little bit harder to pull off at the moment. But we'll be coming soon.
No, that's great, and great to know. Still on ongoing development. Next question. Does Translator utilize the knowledge portals being developed by the Broad Institute?
We have membership from the Broad, so there are a couple of investigators from the Broad. Jason Flanick is one of them. We do ingest some data from there, although which specific knowledge portals I would have to look into a little bit more.
All right.
But happy to follow up on that if you have a specific question.
That's great. Possibly last question. I'll defer to Amanda on this. Oh, 2 more came through chat. We'll see how many we can get through. Appreciate your thorough answers, Chris. How are you thinking about bring your own data integration capabilities through the system, or is this being left to a user post-download?
This is a really, really interesting question. It is on the roadmap, but it's not a very immediate on the roadmap, for what it's worth. I think that there are a lot of ideas for how to do it. I don't think we've crystallized on anything yet. One way that is relatively straightforward to do would be, let's say that we've got this series of knowledge providers. It's not too hard to stand up your own knowledge provider. And so at that point, it becomes a matter of, can we just say, all right, you stand up your own knowledge provider and then do the right wiring. And now the reasoners can see your provider as part of the system of providers and use that information in answering questions, right? So that's one approach. It takes a little bit of like, okay, how does that all work? Because you don't necessarily want just any old person sticking their not very good data and polluting your system, but you do want people to use their own stuff. The other aspect of thinking about use your own data is, is the data that people want to use with Translator, is it private data? What are the sensitivities around it? Do they need to bring information into their own system where they have it? The other kind of like more wackadoodle idea that we've talked about are more like, well, maybe the right integration level is at the LLM level, right? So you pull a bunch of structured information out of the graph, and if what people are talking about wanting to integrate with is text, like I know these papers and I like this information. Tell me about how it fits with, this structured knowledge. Maybe that's an LLM level concern.
No, great answers. Thanks, Chris. This will be our last question, and we'll try and keep it short so we can hand it back to Amanda for her announcements. Is the NCATS translator useful if used to highlight dynamic relationships between genes, chemicals, and diseases in educational modules? Meaning also, is it fully validated?
Fully validated is a good question. It is, I would say, I don't even quite know what fully validated would mean, unfortunately, right? So we ingest a lot of sources. We do apply quality control, but there definitely are a range of sources in them that range from high recall, low precision things, like some text mining results, to much more controlled, structured information. And so this is a lot of why we are so adamant about providing the provenance of edges to the user, because we know there's some clinkers in there, right? And so it's important for us to be able to show somebody, here's a thing we think, but you should go check on this part yourself.
Oh, great. And thanks so much, Chris.
Absolutely. Happy to do it. Really enjoyed it.
And as I hand it back to Amanda, I'll just call out that Chris's email is on the screen for all of you if there are questions you didn't get to.
Thanks, Becky. And we will be including that in our wrap-up materials as well as the recording. So folks, you will have everything you need to move forward, hopefully join the Ambassadors and Champions program or spread the word about that. Yeah, thank you. I just want to take a quick moment to run through a couple of our upcoming programs. My colleague Leah is going to help me pop through these slides real quick. Just some visuals for you guys. We have an upcoming Intro to Generative AI for Coders. We want to move through these relatively quickly. That's okay. The big call to action will be to invite you guys to sign up for NCDS newsletters so you can hear about all of these amazing programs that are happening just about every month. We love bringing in speakers like Chris, and if you have an idea for a program or development that would make a really exciting DataBytes event, please feel free to email me. I will have my email included in our wrap-up materials as well. But we do ask that you sign up for our newsletter, and yeah, feel free to reach out about any comments or feedback you might have. And we look forward to seeing you at the next NCDS program and potentially at the next RENCI event in the very near future. All right. Thanks so much, everyone.